Hi, I'm Isha,
And I'm Drew.
and today we're going to be talking about our work with Drosophila melanogaster, or more commonly known as the fruit fly.
Let's start by looking at our different unknown mutants. Our wild type had a ruby red eye color with centralized black dot, while the opt mutant had a uniform white eye color. Finally, the gum mutant had an entirely black eye color. Moving on, our primary focus was on ensuring the purity of the genetic lines by isolating virgin females from our fly cultures. Using virgin females is a critical step as it ensures all the offspring come from the same genotype father. This is how we did it. We began by selecting virgin females from fly vials aged 7 to 10 days, a period marked by numerous dark pupae. This timing is critical for ensuring that we're working with flies at the right stage of development. To facilitate the collection of virgins, all live flies were removed from the vials 6 hours before our collection time, leaving only the pupae. Upon hatching, we put the flies to sleep with ether to ease the sexing process. It's important to note that we use sex chromosomes as the sole criteria for determining gender. This allowed us to accurately identify and isolate the virgin females while disregarding the males. After this, the vials were cleared again, leaving the remaining pupae for an additional 6 hours before our final collection. By choosing females for the mutant strains for our crosses, we significantly enhance our chances of detecting any recombination events. Now, let's look at the specific crosses we conducted. Our first cross involved mating wild-type males with virgin females from the gum mutant strain. Our second cross involved mating wild-type males with virgin females from the opt mutant strain.
Our first cross was between a female fly with a recessive gum mutation and a male wild-type fly. As seen in Table 1, all the resulting flies had the wild-type gene, with there being 32 female flies and 33 male flies. Our second cross was between a female fly with the opt mutation and a male wild-type fly. Table 2 shows that all 37 of the resulting male flies had the opt mutation, while the 43 resulting female flies were wild-type. Because we knew that whichever the autosomal mutant was would be recessive, we predicted that the autosomal mutant crossed with the wild-type male fly would result in all the offspring being wild-type. And because of this, we knew that the gum mutation was the autosomal mutation. Because gum was the autosomal mutation, we knew that opt had to be the X-linked mutation, but we did the second cross to affirm our beliefs. All male flies have to get their Y chromosome from the male parent, and the male flies in the parent generation are wild type, so they cannot have the mutation on their X chromosome. Male flies also need to get their X chromosome from the female parent. Because the female parents are all mutants, all the male flies would receive an X chromosome with the mutation, and therefore would need to have the mutant phenotype if this is, as predicted, an X-linked mutation. All the females, on the other hand, would have the wild-type phenotype because the male parents are not carriers, so they would receive a wild-type allele from the X chromosome donated by the male parent and a mutated allele from the X chromosome donated by the female parent. If the mutation is X-linked recessive as predicted, this would result in a heterozygous dominant genotype or a wild-type phenotype. This was the case for our cross with opt, proving that opt was the X-linked mutation.
Now, let's step back and look at 3-point mapping, where we would have chosen the gum mutation. This autosomal mutation provided us with an opportunity to explore genetic interactions and distances. This is how we would have done it. First, we'd start with the F1 generation characterized by the genotype LY plus SB over plus U plus. In this notation, U represents the unknown gum mutation. This initial setup was critical for our mapping process. The next step would be to cross the F1 flies with the genotype of plus U plus over plus U plus. This cross was designed to produce the F2 generation from which we could gather vital genetic data. The F2 generation is then categorized into 3 groups: non-crossovers, single crossovers, and double crossovers. With our categorized data, we then could calculate recombination frequencies. These frequencies are key to estimating the distances between the genes in question. We determined that the gene pair with the highest recombination frequency was the furthest apart. This gave us insights into the spatial arrangement of these genes on the chromosome. The least common class of offspring involving the middle gene flipping indicated the gene order.
Because we were not able to gather this F2 data ourselves, we were given simulated data as shown in the table. And using the same 3-point mapping procedure that Drew talked about previously, we were able to map the location of our mutation in relation to the LY and SB genes. We performed a 3-point cross, determined that the LY gene was in the middle, and then using percentages of recombination frequencies, found that the mutant was 12.5 map units from the LY gene and the SB was 16.5 mutants from LY. The labeled map and simulated data are shown in the slide. The hypothesis generated was that our mutant was in the sepia gene. We knew that our unknown mutant was on chromosome 3, along with LY and SB genes. So after determining the map described in the last slide, we found LY and SB in the chromosome map provided on the screen. LY and SB are boxed in red. Knowing that the mutant was 12.5 map units from LY, but before SB in the chromosome, we determined that the sepia gene best met the criteria. The sepia gene is also boxed in red on the chromosome map. To prove once more that the mutant was in the sepia gene, we compared the phenotypes of our flies with the gum mutant to the phenotypes of flies with the mutant in the sepia gene, as pictured on the slide, and found that the phenotypic change was the same or very similar. Therefore, we strongly believe that our mutant gum is in the sepia gene. To further prove that this is the case, a complementation test can be performed, though we did not do so. Both mutations are autosomal recessive, the mutation in the sepia gene and our unknown gum mutation, allowing for a complementation test to be possible. One fly with the gum mutant can be crossed with another fly with a mutation in the sepia gene. If the mutations are on the same gene, then the resulting offspring will have the mutant phenotype with black eyes because both the recessive alleles necessary to result in this phenotype are present on the same gene. If the mutations are not on the same gene, the resulting offspring will have the wild type phenotype with red eyes and a centralized black dot, because in both genes there will be one mutant allele and one wild type allele, and the wild type allele is dominant over the mutant allele.
We were informed by our professor that the gum mutation is located on chromosome 3. Through genetic mapping, we were able to calculate its position relative to the known genes on the chromosome. It's approximately 12.5 map units away from the LY gene and 29 map units away from the SB gene. Notably, it's positioned closer to LY than to SB. Our hypothesis zeroes in on the sepia SE gene, which is situated at 26 map units on the chromosome. This places it 14.5 map units from LY and 32.2 map units from SB. Because these are very similar distances to the predictions made on the gene map, we can conclude the gum mutation has a high possibility of being on the sepia gene. Looking ahead to future research, our focus could shift to understanding the specific impact of the gum mutation on the sepia gene, particularly its role in drossopterin synthesis. Drossopterin is a key pigment influencing eye color, as noted in FlyBase. Our objective would be to delve deep into how this mutation affects protein expression linked to this pigment synthesis, thereby affecting eye color. Here's how we would do it. We'd employ a Western blot analysis as our primary tool. This technique will allow us to compare protein extracts from the gum mutants with those from the wild-type flies. Our focus would be specifically on proteins involved in pigment synthesis pathways. Then, we'd anticipate the gum mutation will lead to noticeable changes in the regulation of proteins encoded by the sepia gene. This could manifest as either an increase or decrease in protein levels. Additionally, we might observe alterations in the protein structure. These changes would be expected to be evident in the Western blot as variations in the intensity and size of the protein bands. To ensure the accuracy of our Western blot, we will have to include two types of controls. A positive control will be a protein known to be affected by the mutation. Its presence will confirm the effectiveness of our Western blot. And a negative control will be a protein unaffected by the mutation. Its inclusion is crucial to ensure that any changes we observe are specific to the proteins of interest and not due to nonspecific interactions.
Thank you.
Thank you.